PCI positive, go to compound, such and such. So we all pretty much use the same methods for that. Now I'm going to hand it over to Sarah to talk to LCMS.
We have the LCMS results of the NIMBAC, where we were able to identify 3 molecules, which is the nimbin, guanine, and azidorin. And then we have the LCMS results for the NIMLIS. We were able to identify the nimbolide, guanine, nibinene, and epoxazadirodian, that one. So from the true results for the LCMS, for that of the NIMLIS and the back, you can, there are some molecules that are similar in both the results. So these are the LCMS results for the endophytes, where we were able to identify epicatechin in the LCMS results for the back and the NIMLIS. So for the endophytes, there is from what we found, it seems for the endophytes we have a similar compound that is in both the back and the leaves. Then this the LCMS results still for the endophytes. We were able to get the nibinin, epoxazadirodion, and the nibolide, which are also similar to the LCMS results for the leaves. Then the GCMS results for the NIMBAC. We just identified a few, 4 molecules, but there were more, which is not displayed in our slides. And that's the GCMS results for the NIMLIS. We were also able to identify more, which we also didn't combine it in our slides. We just identified just a few, like 6 of them or 7. Then for the GCMS results for the leaf endophytes, we were able to identify more than 10 molecules in the pigs. Then that's for the leaves, and then for the back endophytes, when you compare both the back and the leaves endophytes, we are able to identify that for the leaves endophytes, we have more molecules than the back endophytes. Yeah.
Okay, so in terms of our HPTLC results, what we did in terms of categorizing our results, because we had so many samples with the leaves, the barks, and the commercial products, we put them together by sample and then with our low, medium, and high polarity. So I'm going to skip over, because we're running out of time, to our 366 nanometer development. Our first sample is our bark for our low polarity. We had a lot of terpenes in our samples for both the neem bark and the leaves, and so all of our low polarity samples were eluted very distinctly, and we think that's because of the terpenes. In the leaves specifically as well, I'm going to run over here and show you. This is our low polarity and our medium polarity for our leaves. Our leaves also had a lot of medium polarity compounds in it as well, and so that was reflected in our HPTLC results. Our commercial products on our far right side also showed some conservation of specifically our leaf compounds that were in our medium polarity solutions as well. And here that's also reflected in our derivatization samples, and we can see as well under our RT white light that we had a lot of low polarity in the bark, a lot of low and medium polarities elution in our leaves. Our final thing that we did was an antioxidant image. Our epoxy compound is known to be an antioxidant, and so if you look at our white light samples, especially for the bark, we see a lot of white in that sample. That indicates that there is an antioxidant present. In our leaf, we see it right here by where our sample was placed, a little bit further up. And that was also reflected in our 366 nanometer development, which looks for bioactivity, which is also often associated with antioxidants. So, in conclusion, because we are running out of time, neem leaves and bark contained a lot of secondary metabolites that had anti-cancer, anti-inflammatory, insecticidal, antifungal, and antibacterial characteristics. So it's a great plant. We also found that the endophytes may promote the production of these metabolites as a product of their symbiotic relationship with the plant. And the leaves and the bark do appear to promote significant antioxidant activity. So in the future, what we would like to do is use standards of the molecules from our molecular compound library, and compare that to our results, just so that we can more assuredly and decisively say that we did have these compounds in our HPCLC results. We also want to analyze the roots, the seeds, and the flowers, because those parts of the plant also had significant compounds that were very beneficial medicinally. And thank you very much. That's it.
Oh, that was very nice. As I told you, at least I've seen 5 people sleeping. Please avoid that. Because next time we'll be the one, we'll stop the presentation for you to address us. The talk was really nice. Maybe I'll just start off. So what is new in your work?
Pardon?
What is new?
What is new?
Yeah.
All of it? Okay, so specifically the endophyte data, I don't think has been done recently. And so in terms of the endophyte data, we found this epicustachin, which is actually an antioxidant compound. And so that, to me, suggests that the antioxidant assay that we saw in our HPTLC may be attributed to this molecule that we found in both our bark and our leaves.
Now why I ask you is because neem has been studied all since we studied natural products. At least I'm happy you picked up the endophyte data, though one of the compounds that you reported, the cyclotrisiloxane in the bark.
In the bark?
Yeah.
For GCMs?
Okay. So this compound, we all agree that this is from the colon.
Yeah.
Yeah. So that is not a compound that you need to report. So maybe now next is to get enough concentration for the endophyte, try and do some really cool science, pin some new maybe biological significance and importance to it, and voila, we have something to present. So I'm going to the floor. Anybody to, okay.
Yeah, thank you. That was a very good presentation. I liked it. And my question is, how do you differentiate when you look at the compounds from the endophytes and the secondary metabolites from the plant? How are you able to tell that this particular compound is synthesized by the endophytes and not the plant itself?
Okay, so we analyzed the endophyte data separately from the data with the leaves and the bark. We looked at abundance, we looked at peak level, and so when we were looking at our endophyte data, we found this specific compound that was not very much so abundant in just the leaf and the bark samples. And so that's what makes us believe that the endophytes are significantly contributing to the production of this compound.
Scientifically, the way you could have approached it is maybe using a labeled molecules, whereby you infuse labeled carbon into the plant, and then if you're able to pick it up, let's say maybe I see that if you're able to pick it up in endophytes, then you could say this synthetic pathway starts from the plant. But if you're not able to pick it up in the endophytes, then you say it ended up in the plant. Okay, so those are things that you need to think of when you're doing your future studies. Unless you want to add something. Yeah.
So one thing I want to remind you, one place you said the molecular weight was derived from more. So when you do past never used that's considered natural. It uses apple leaves, isotopy method, not molecular leaves. So the molecular is an average. It's not applicable to mass spectrometry.
Yeah, well, that's fine. Also, your structures are too tiny. See, somebody sitting at the back, just seeing like a dot. Remember, we are training you to go to big stage to give presentation. So the less is more. Remember that, yeah? It's easier just have 4 structures, chemical structures, and then maybe just a name. Then the anti-malaria or antiviral or something. You just stop that. You gotta try it. Any other question from the floor? If not, we give them a head clap. Head clap. Not clap, head clap. Let's shake your head. Okay, that's nice. So I want to welcome the next group. Melia.
Hi everyone, I'm Charles Obiambo.
I'm Miyamo.
Justin Mutiza. Yeah, so we worked on Melia Zidaranga. So the first one we have the Melia Zidaranga. It belongs to Melissae family and their benefits. So the introduction part of it, we have characteristics, commonly known as China berry tree. It is a native in some part of Asia, such as India, Pakistan, and China, and invasive in African countries such as Tanzania and Kenya. Melia zidaranga is 6 to 35 meters in height. Then leaves are bipinnate, flowers are pink to lilac and star-shaped, about 18 millimeters in diameter. Fruits are small, yellow drupe, nearly round, and about 15 millimeters in diameter. Then we have some of the compounds that we found when we were doing research on literature, part of it. So those are the compounds that we found while doing research on literature part of it. Then we have the application, that is the overall chemicals profiles of Melia zidaranga contribute to its biological activities such as traditional medicine and natural insecticides. Then we have Melia zidaranga leaves, nimulin, which offer antimicrobial, anti-inflammatories, anti-stigmatic, antioxidant, and anti-cancer. Then we have the flowers and the leaves are applied as poultices in the treatment of neuralgia and nervous headache. Then the stem bark is anthelmintic, astringent, and bitter tonic. Root bark is emetic, vingative, and vermic. The seed is antihamic. Insecticides, we have the aqueous and the alcoholic extract of leaves and seeds reportedly control many insects and mite and nematode pests. Fruits produce the chemically related malicapines and that have acidarantine-like bioactive in many types of insects. The leaves repels mosquitoes and other insects. Moreover, contain a unique limonite melatonin that has both antifungal and insecticidal activity against a range of insects. Objectives. Our main objective was to identify and quantify metabolomic constituents of Amelia acidoranth leaves and bark, compare the compound with existing literature and determine their benefits. Then we have materials and method where we have the sample preparation. So first of all, we have to label the sample. Then we weigh 200 mg. Then we have to add around 1,000 ml microliters of methanol into the vials of HPLC. Rotate for 5 times and put the sample into the sonicator for 15 minutes. Centrifuge at around 3,000 RPM, that is the speed, for 15 minutes. Then we filter the supernatant using power tools to collect 0.5 ml the filtrate. Sample were labeled and processed using appropriate machine. Then we have sample processing method where we have the leaves and the bark sample collected from machacos and then dried, cast in fine powder. So it is in form of a flow chart. Then we have under LCMSC we weigh 200 mg, then we dissolve in 1,000 microliters of methanol. Then on the GCMS and HPTLC, we weigh 200 mg, then we dissolve in dichloromethane. Then data processing, we have the raw GCMS, LCMS data. Then we have chromatogram conversion, mass spectrum evolution. Then we had to identify the peaks, quality, retention time. Then we had to match the chromatograms. Then visual and analyze the cover data and generate the exact forms. Then we have under HPTLC. So HPTLC is whereby we quantify or we can identify the compounds. So the first step was the application. So the application is whereby we were doing the spotting. So before we do the spotting, we had to draw the plates. Then we measure the baseline and the solvent front. Then we do our spotting. That is the first step. Then under the development, that is where we have the chamber. So the chamber is whereby after preparing the mobile phase, we leave it to equilibrate for around 15 minutes. Then we put our sample so that it can run with the mobile phase under through the process of capillarity action. Then after that, we'll be able to remove our plate after it has reached the solvent front. Then we dry it up for the spotting. Then we have the next step is derivatization. That is where we were using some of the reagents such as CPPH, radicals, or sulfuric acid, so that we can be able to identify the antioxidant compounds. Then we have the documentation. That is whereby after we obtain our spots, then we were now able to document the results. Then we have to process the analysis.
I'm going to take you through the results that we got. And using GCMS, we found 14 compounds, the highest being vitamin E and the lowest being capesterol. And still on the back using GCMS, we found 8 compounds, vitamin E being the highest and the lowest being diphenhydramine. And we tried to compare the compounds that we found from the back and the leaf, and we found that the leaf and the highest number of compounds, that's 14, and the back had 8 compounds. But there was similarity, like the vitamin E was found both in bark and leaf, and squalene were found both in bark and leaf. On the LCMS, based on the libraries that we got, we were able to search the compound that we found, and we found routine as one of the compounds in GCMS, and we also found theory design based on the library that we already found recently. And still on the same leaf, we are able to find a chloropyramide based on the molecular mass that we found. And still on the back, we are able to find a transofluronic acid, which was similar on the leaf, which matched with the one we found the leaf, this one. So it still matched on the back using the LCMS. And on the endophytes, we found there was no similarity in the compound that we found. The highest compound was N-phenyl acetate, but then did not match on the compound that we found from the leaf and the back of the plant. On antioxidant properties, we were able to find that there was high antioxidant properties in the back than in leaf. Just like the neem, they were able to find that there was high antioxidant activities in the back than in the leaf. And we are able to get the molecular profile of the compound that we found. Using the HPLC, the different polarities of the developing compound, we are able to identify different compounds and the different visualization, 254 and R366. And I'll take my friend to take on the application of the results.
So, just to give a brief background, metabolomics fits in the study of multi-omic approaches, along with glycomics, which is the study of the glycome, epigenomics, which is the study of the epigenome, lipidomics, which is the study of lipids, genomics, the study of the genome, and transcriptomics, which is the study of transcriptional and translational processes, proteomics, which studies proteins, and then glycomics, which studies the glycome. So to begin the discussion, as he said before, there were 14 compounds discovered with relatively high abundances. For the leaf samples, those included vitamin E, followed by squalene, which showed the highest percentage in abundance. All of these compounds share antioxidant properties. So we concluded that this contributes to protection against reactive oxidative species, which is ROS. It contributes to mitochondrial membrane potential dysfunction, inhibition of lipid peroxidation, hydroxyl superoxide anion, and nitric oxide radicals. For the bark samples, vitamin E displayed the highest percentage in abundance, followed by gamma-citrullosterol, contributing to the bioactivity properties such as anti-hyperglycemic and anti-hyperlipidemic activities. Vitamin E, which is the triple R alpha tocopherol, showed antioxidant properties as well as it protects the cells from free radicals. Also contributes medicinally to cancer risk reduction, eye and heart health protection, as well as immune system function by maintaining healthy blood vessels. And then squalene, which is a triterpenoid or hydrocarbon, displayed antioxidant and emollient properties as well. So all of these contribute to protection against radicals and reactive oxidative species. This is just a briefer background of the medicinal properties of the 14 compounds found. So to continue the discussion of metabolomic analysis, LCMS confirmed that the compounds 2-transylferroglycuric acid contributes to the inhibition of reactive oxidative species by exhibiting antioxidant properties in vitro. Routine inhibits osteoclast formation by decreasing reactive oxidative species, and TNF-alpha by inhibiting activation of NF-κB. And chloropropamide exhibits anti-aging effects that depend on mitochondrial complex 2 and the production of mitochondrial reactive oxygen species. The discussion for the HPTLC also confirmed that more antioxidants were present in the leaves that are right than the bark. And according to the results of the antioxidant assay, these compounds do not correlate with those that were discovered in the GCMS analysis. To conclude, according to the compound library prepared prior to the presentation, 4 of the 11 compounds matched the original compound library. These compounds include routine, which was present in the leaf sample, 2-transylferroglycuric glucaric acid, which was present in leaf and bark, thiouridazine, which was present in the leaf sample, and chloropropamide, which was also present in the leaf sample. As stated before, they all exhibit antioxidative, anti-inflammatory, antiviral, antimalarial, antiprotozoal, antihypertensive, and anti-ulcer properties. The metabolomic analysis, which includes GCMS, LCMS, and HPTLC, confirmed that these compounds contribute to the inhibition of reactive oxidative species. And that's just kind of repeating the same information that I already said. So the future directions for this research includes emphasizing replicates in areas that were not performed correctly, which were collected initially. Also extracting samples from different locations that grow the sample natively and non-natively, and then expanding the endophytic data for our plant samples. These are our acknowledgements and our references.
Wow, thank you. Give them a traditional clap. It was really nice. I wish to know how long did they take to finish up their presentation?
14 minutes or 20 minutes.
Okay, they're still within the time. But when you go to maybe other platforms, when you're given 10 minutes, somebody will come and snatch the microphone from you. It won't be so nice. So when you see somebody pulling up a placard, don't now start speaking fast because you'll confuse the audience more. Just summarize whatever you are. You're not supposed to go through the whole slides, okay? Just quick one. The three of you did well in talking, but you didn't do well in getting to us because you're all talking to the board, yeah? If you are keen to notice that, you're all reading the slides the way they are. That's why it's discouraging people from writing sentence by sentence when you are, because you'll be tempted when the stage becomes so hot, you'll be tempted to go and start reading whatever you've written down. So it's easier just put point form that you can now talk about when you're working out. Next, I want to talk about there's some few maybe just talking error or something. Somebody said, one of you said 1,000 ml microliter. We don't have such like volumes in terms of measurement, okay? Those liters. Maybe another question to you is, how do you use sulfuric acid to determine the antioxidant properties of inactivities? Because you mentioned it out. Like, you guys, they all go, somebody can. Yeah. Okay. So just write it out. Yeah? If you don't have the what is it called, the normal visualization, still in the other the traditional TLC place, we normally use sulfuric acid to just do visualization. We spot it up and then burn the laser. And then for HPTLC, I can just elaborate it out.
Yes, I think as you remember, we have done two types of differentiation. One of them is with sulfuric acid. I think this is the one that you meant. And this to visualize some compounds that doesn't have any activity, like for example sugars or amino acids. And for testing antioxidants, we have used the DBPH solution. So this is two different solutions, totally different ones.
So last from my point is protein. From your slide, you said very well it came from LCMS. And then you're telling us, you're lying to us that it came from GCMS. Why? Your written is from LCMS, and then you're telling us it came from GC. Okay, what about that? Try and understand whatever you've written now. So I will take 3 questions from the floor.
That was a wonderful presentation. Thank you so much. Mine isn't a question. It's just a comment. On your slide on sample preparation, you said that dechloromethane extraction was both for GCMS and HPTLC, which is not right.
So what did you use for HPTLC?
Methanol.
Same extract like in CMS. Yeah.
Thank you so much for the wonderful presentation, and I think I also have a comment on one of the slides during the discussion that the font was so tiny, we couldn't then and crowded, tiny and crowded. Then maybe it's good to try and not to put too many words on your slides. It tires the people that are watching. Then the other one, I'm just asking, probably out of ignorance. Now, when you're working on endophyte cell, I'm told, I know there are fungi or bacteria that grows within the plant. So for you to screen them for the metabolites that they produce, do you first of all extract the endophytes from the plants, or how do you go about it?
I can probably answer that one. So currently I am doing endophytic research currently with both the Melia and neem. And so the process that we do is we cut up the samples, then we sterilize them, first 95, then 10% bleach, and then finally we do 70% for 2 minutes. And so that's surface sterilization, and we put those on a medium. Right now we currently have 2 mediums, MEA, which is malt, and then the second one is potato starch. So now currently we grow the fungi and the bacteria through leaves, barks, and resin. So that's how we culture them.
It's because they dump that, so I think they are sealed.
Yes.
We didn't get that information.
Okay, that was really, at least thank you for clarifying. I want us to give them a round of applause. They really did a wonderful job. Something interesting, I think we are growing more and more scientific over the years, because people who are here during the first year, they have seen tremendous growth. For the second year, this is just a wonderful, in terms of presentation and everything. So I wish to welcome right now the next group, which is Kenya.
Go here.
Hi everyone, we are the Family Somnifera team, and my name is Melissa Mingo, and this is Ian Wonky. We're happy to be up here today, and without further ado, our introduction.
All protocols observed. Good afternoon. So, first of all, what is with Daniel Somnifera? It comes from the family of Solanaceae, nightshades, and it leaves smells like green potatoes, and sometimes it smells like wet horses or horse sweat. It has alternate leaves, which are opposite and to the flowering stalk and covered with silvery hair. Usually grows fast in full sun, alkaline and sunny soil. Propagation is done through seeds, or stem cuttings. So mostly of its mechanism through the research, it has anti-inflammatory properties which occur by inhibiting the IL-2, IL-6 and IL-8 proteins, also by inhibiting the action of NF-κB and decreasing the COX-O2. It also has a cardioprotective, neuroprotective, antidepressant, anti-anxiety, anti-diabetic, anti-epileptic, hepatoprotective properties too. Also, it inhibits the HSP70 and HSP90, and also the MPRO, which is involved in enzyme carcinosine. Complication due to overusing it is dependency, and also acts as an immunosuppressant. So, to the methodology, our study area was in the UN and also ISIPA chemistry laboratories. Method of collection was purposive. Sample types, leaves, and so the methodology, extraction, first we sample collection, then fine grinding of the parts of the plant that we were going to use, and then obtaining 2 grams of the fine grinds of the part and extracting through methanol. That's for HPLC and LC extraction, and also for HPTLC And then the next thing was sonication, followed by centrifugation, filtration, and then inserting into the HPLC and LCMS machines. Then separation occurs and you do your analysis. For the GC, the only difference was the extraction method, where we used the dichloromethane. Yeah, and for HPTLC, there are five steps. That is application, which we did using the Leomat 5, and then in the development chamber where we used 3 mobile phases. One is the low polar, dissolving solvent. Next was the medium polar, high polar, and our derivatization reagent was anisaldehyde sulfuric acid. So to the results, as you can see, this is what we obtained for library. That's for the endophytes under different light. That's for the leaves and roots for different mobile phases. Yeah, and that's for the stem. Yeah, so I'll give it to.
Alright, thank you very much. I'll be taking the rest of the results. And first I'm going to talk about LC, and then I'm going to talk about GC. Now, we weighted our LC analysis a little bit less heavily in our discussion, and you might ask why was that? Because our LC didn't turn out as well as we wanted it to. Our expected compounds were very, very small in concentration, and you can see like we had to zoom in to picture it. And for this reason, and also because like everything we found was already in our molecular library, not that interesting, I'm going to speed through a little bit. It was kind of expected. We got a lot of withanolides, a lot of alkaloids, which is par for the course with somnifera, and yeah. So this is kind of a table of everything we found. This combined with withaferin A and with these withanolides are.
Hate to interrupt you, Liz, but unfortunately Dr. Ahab has his flight to catch, but if we could all just give a heartfelt thank you for all the work he's done, because our analysis would have not been possible without him. So, round of applause for Dr. Ahab. Continue.
Alright, so we have this table, mostly expected. The withanolides and ashwagandha in particular, those are some of the most bioactive that we have found. Now, our gas chromatography results were more interesting. And I know people are going to complain that the font is small. I will send you the slides if you're interested. Interestingly enough, we found not just antioxidant things, we also found a decent number of toxins. So for example, this one, which is alpha-copain, it is associated with autism and Crohn's disease. The carfilin oxide, which I actually think Tony's team also found, it has a wild list of flavoring, fragrance, risk of chromosomal defects, brain and stomach and lung neoplasms. The list goes on. It's pretty toxic. But then at the same time, we also have squalene and these phytosterols, some of which raise cholesterol, others lower it. But we've got a very mixed bag, which is kind of what is kind of expected. And I'll explain that in a bit. And so this is the fit figure for our leaf GC. Now, for root, this was probably the spiciest one. Because we found tropococaine in our plant, which I thought was cool. Because it is a member of it's kind of like cocaine, except 50% as effective. All natural, I guess. So there's that. And we're excited to have that. It also does it also does fit into the risk of dependence, interestingly enough. So I'm wondering if that's somewhat in here. Interestingly enough, we didn't actually see any opioids, even though it has been reported in ashwagandha, but that's not something we saw. We also got, yeah. All right. And we still have alpha-pinene and like carotene, and yeah, some plasticizing agents, that sort of thing. Okay. And finally, we have the endophytes. Now, the endophytes were interesting because we only got 4 compounds. And they don't seem to be super interesting compounds if you look at it. We've got propylene isobutyl acetate, which are every kind of metabolite. Everything uses these at some point. And so it's not that interesting. What we did find are like epoxides and this thing, which are definitely associated with diseases. Okay. So, I'm going to walk you through it. This is what we like to call a radar chart, and it's exactly like a line graph, but it's wrapped around. And so it's kind of like a combination of a Venn diagram and a pie chart. Okay. So over here we have the leaf, and you can see it has the highest beta carotene, and then at almost 75%. Because this is 75, this is 50, this is 25. It's got lowest of germacrone B. You can see that each tissue is split up, and this split is probably the most interesting thing we found. Because this is not a random order, it's actually ordered by polarity. So I was not expecting this to be a thing. I just wanted to make sure that it was ordered by molecule and not by tissue. They ended up grouping themselves together, even so. If these results are replicated, it does kind of suggest that that polarity is another characteristic that should be reported about tissues, along with, say, acidity. This could be important, say, in like drug development, for example. Okay. In conclusion, with somniferous bioactives really different from its endophytes. And so endophytes were not producing them. Each tissue and organism differed by polarity. Contamination is pretty likely, like we saw in the LC. And this is even more likely because we did not have standards and we did not have replicates. Thus, we need the replication to make sure that my nice graph is true. But many compounds did still match the molecular library. The mix of toxins and antioxidants made a lot of sense because many people report very different effects when they take ashwagandha. And so it raises the potential for fluctuations in that causing different reactions. And these are acknowledgements, and this is our weekly application.
Okay, thank you. Give them a round of applause. How did they do time-wise?
Like 10 minutes, 10 seconds.
Well, that's really nice. Yeah, so at least we have 5 minutes for questions. Anyone from the floor before I wrap up? Was the talk so clear that I can see you?
So you see some compound have toxicity.
Mm-hmm.
Also, ashwagandha is used as in India called India ginseng.
Yes.
So it's reported for some side effect.
Mm-hmm.
So can you see any compounds correlated to this side effect?
We saw a few compounds that were correlated to various side effects, but most of what we saw that was labeled as toxic was like irritant or carcinogenic or mutagenic. And so I'm sure that many of them also cause these side effects in some way, shape, or form. But when you're looking at PubChem and it says acute contact dermatitis, you have to ask the question, well, at what concentration? And so it's very possible that something that causes cancer in like a lab concentration could cause whatever side effects people report in a lower one with combination of the other molecules.
So in addition, did you see some different for this toxic molecule in the root and the area?
Yeah, I did. In fact, all of our tissues differed pretty substantially. We had a couple instances of alpha-pinene. We had our sterols popping up with regularity, but pretty much everything else was different. The root was the most toxic, And you can see that like there's not only tropococaine, but also tropium, as in the precursor to atropine. There were a lot of ACHE inhibitors, and so those are potential medicines, but it's also very commonly used as a poison. And so, yeah, they were a little spicy. Many of them were actually associated with Crohn's and digestive issues and like autism, which was kind of interesting.
Okay, that was really cool. So, assume I give you half a year. What different will you do for this particular project?
Oh, okay. I got you. So, first of all, I would make sure that we had standards so that we could, to run on our GC and LC machines, so that I knew that for a fact that what we're reporting as M-xylene is M-xylene, and that's how it appears on that GC machine. Second of all, I would do a lot of more replicates. 3 is kind of standard. I'd like to do more because it adds rigor. And the other thing is I found the polarity difference kind of the most interesting. This kind of happened by accident, and I haven't really looked at it much further than, I think that this is probably an important factor to note about tissues. But I would love to dig more into this and find all the implications, because I'm sure that there are a lot.
That's really nice. So when you take it, this particular data, right now you already know that yes, it has all this particular compound. So what?
I'm sorry, could you repeat that?
You have identified all these particular compounds.
Yes.
So what I'm asking is now for your future outlook, not repeating the experiments. Maybe
So you're not asking me what I would improve?
Yeah.
Okay, so I guess the implication is that ashwagandha really needs to be better regulated, and that it needs to be a really strictly controlled medicine. And that's because of the mix of toxins and medications that we found. It does have antidepressant and anti-anxiety activity. People tend to think of antidepressants as a stereochemical and antidepressant plants as just fine. This is not just fine. We probably need a prescription.
Yes, that's true. I really like that. Maybe just a comment is, if I tell you my height is 1 times 10, maybe negative 2, does that make any sense? The same way to your areas. Please put a scale, okay? The peak areas.
Yeah, there is a scale. See? So this is a percent area. This ring is 25%. This ring is 50, and then this ring is 75. We didn't have anything higher than that.
Yeah, now go back to the other table.
Yeah. Okay. And so I think that these peak areas. What's
Let's
happening?
go back.
Yeah. This peak area is already adjusted, I think, for percentage. And so this is.
Just pick it up. Go to the other table.
Sure. And then this right here, this is peak area.
Yeah, so do they make any sense? That's why I'm saying you need to be consistent in terms of your presentation. So that when somebody looks through your data, they quickly identify that yes, it is 4%, it is 10%. This is the most abundant component. So.
Yeah.
But overall, you did well. Try and improve on your slides. Don't tell us that you're going to give us later on after presentation. And
How about environmental influences? Did you speak to this, or maybe something that we do?
I didn't, because that's kind of beyond the scope of what I was looking at. If you wanted to do something about the environmental impacts, I would suggest going on PubChem and looking through all of these compounds, because that would be an important thing to do before making it like a regulated medication. Because one of the big issues, just globally, is that once you take meds, it goes into the water system. It does not just all get filtered out in the kidneys, kind of by design. That would be death, right? And so we don't have necessarily a lot of filters, even in America, that will pick up on like medications such as opioids and whatever have you. So you would want to look into that.
your plan, did it pick that particular, the opioids from the environment or the medical?
We didn't register any opioids.
Around the where you collected the plants from?
But we know that in the literature it is reported, and that's cited as a reason why it might induce dependence.
Okay, any other question from the floor?
Yeah, so that environmental issues is what we say is the angle I was talking about. The fact that there's also some of those ingredients could vary according to where you get the plant.
Yes, yes. In order to do something like that, we would need to collect multiple samples from multiple regions and do a lot of replicates.
Yeah, that's what some of the things we can do in the next round.
Yeah. Absolutely.
So please don't follow that. If you want to get high, don't see that you found something.
Yeah, don't steal our tropical cocaine, please.
It's caution, okay?
It's ours.
So give them a round of applause. Welcome we're almost there. Wow. The aloe vera group.
Okay, good afternoon, everyone. We stand before the existing protocol to present our project titled Metabolic Analysis of Aloe Vera Leaf Extracts. My name is Abokar Esringim, and my colleague, Kenyan colleague, Victor Otieno. So, without wasting your time, I will just carry you through the background, some of the traditional features of aloe vera, and of course this is a plant that is, doesn't need much introduction. It is one of the most popular plants we all know. So it is nicknamed the miracle or wonder plant because of its health, immense health benefits to humans. And this plant has been reported to have over 75 different nutrients and 200 active compounds. The plant has antimicrobial, antiviral, and anti-cancer, and as well as anti-inflammatory properties. And its medicinal values has been applied for over 6,000 years. Extract and gel from aloe vera is being applied to treat so many diseases like burns, like eczema, like headaches and constipation, as well as laxatives. So in this study, our main objective was to analyze the secondary metabolites of aloe vera using leaf extract, using the HPTLC, LGCMS, and LCMS. So just a note on the introduction of aloe vera. The use of aloe vera in traditional medicine traces back to 6,000 years in the ancient Egyptology. And right there is the Ebers Papyrus, which is the oldest document reported on aloe vera, the use of aloe vera in traditional medicine in the world. This is a plant that has a global distribution. It is found in tropical and subtropical regions. Although it was believed to originate from part of Somalia, Sudan, or Arabian Peninsula, it is a plant that has been introduced to many parts of the world. And it contains several metabolites and vitamins, phenolics, and minerals. These are several important compounds and metabolites have been reported on aloe vera, and these are some of the most important and some of the most common compounds found in aloe vera. The aloe, which has anti-inflammatory, antiviral, and anti-parasitic properties. The imodine and the triclosan.
Thank you. I'm going to take you through materials and methods. And aloe vera sample was collected from Karura Forest in Nairobi County. It was dried, then crushed using pestle and mortar. Then the metabolites of aloe vera were characterized via HPLCMS, GCMS, and HPLTMS. Under HPLTMS, preparation was done, we derivatized. Derivatization was through, okay, plates were derivatized by immersion in anilsadehyde reagent, followed by 3 minutes of heating at 100 degrees Celsius. 170 ml of ice-cooled methanol gently and slowly mixed with the 20 ml of acetic acid and 10 ml of sulfuric acid. The mixture was then allowed to cool at room temperature, and 1 ml of methoxybenzaldehyde was added. Okay, HPTLC analysis was done, whereby silica gel glass of HPTLC plates that one, with multiple systems manufactured. Okay, that one is common. The process is summarized in this workflow, where application was done, development, derivatization, documentation, then data processing and analysis. Okay, under GCMS and LCMS methodology, the methods, they look like the procedure is the same. The only difference is that under GCMS, we used anhydrous sodium sulfate at 1 ml of the filtrate to 2 ml glass clear vials. Okay, the parameters were there when conducting the process. Those are some of the photos.
Okay, thank you. So this is the results of our HPTLC analysis, and you can see the different zones and the markers showing the fingerprints of the compounds in the system, in the 254 low polarity, and in the medium polarity, and also the white, and the 366 in the medium, low, medium, and high polarity. And in the figure 6, you can see the separation peaks of different compounds in the aloe vera leaf extract. And this is the results of the GCMS. And this figure 7 shows the abundance of compounds identified from the aloe vera leaf extract from the GCMS. Some of the, let me show you. In total, about 36 compounds were identified. So after sorting and cleaning the data, we remove compounds that have chlorine, hydrocarbons, and silicate. So we ended up with 11 important compounds. And the most important compounds, or the most abundant, were D-amyrin and phytol and vitamin E had the highest abundance among the 11 main essential compounds, while the beta-amyrin and gamma cytosterol being the least. So this is a graph showing the extracted compounds from the aloe vera leaf extract across different peaks in GCMS. You can see the alpha-pinene, alpha-copene, and luteol, and octa-decanal, which were the main essential compounds. And some of these compounds have anti-inflammatory and anti-cancer properties. So this table shows the 11 compounds, the main 11 compounds with their abundances. This figure shows the results of the LCMS positive mode. We identified 4 compounds in the positive mode, that is under the LCMS. And some of these were reported in the literature, like aloimodine, which has anti-cancer effects, and aloresin, which also have properties against hyperpigmentation disorders. So in the LCMS negative mode, we identified only a single compound, which is allosin, which also has antimicrobial and antibacterial properties.
In our discussion, our results identified several metabolites as previously reported. Compounds such as alpha copain and alloimodine are anti-cancer with potentials in fighting against several cancers in human beings. The large amount of vitamin E from the percentage abundance in aloe vera extract have potential benefits for medicine and cosmetics. Although we detect small compounds of alpha copain and tocopherol and gamma cytosterol and octadecanol, more in-depth research could shed more light about the health benefits.
Yeah, and to add to the discussion, one of the compounds that we found and we couldn't find most literature on which it's pyracyl glucosan A. So there is limited research about it, and we were able to detect it in our sample. So in conclusion, the study successfully fingerprints, identified, and quantified various compounds as shown in the aloe vera leaf extract using different approaches, or 3 approaches: the HPTLC, the GCMS, and the LCMS. So identify compounds like alpha-pinene and alpha-copain are anti-cancer, as previously reported by literature. The study could assist in quality control and standardizing aloe vera leaf extract, and we recommend that future studies shed more light on the continuous application of aloe vera metabolites in pharmaceuticals, traditional, and cosmetic industries. So we acknowledge these bodies, the NSF, BOE, Morgan State, and the ICP, and the University of Nairobi. And of course, I celebrate this experience. Thank you.
Thank you so much. So let's clap. I said leg clap. Leg. Oh, that's nice. That was really nice for the aloe vera team. They did a wonderful job. I'll go first and allow maybe 2-3 comments or questions from the floor. Timekeeper, how well did they do?
10 minutes, 50 seconds.
That's still good for the talk. My first comment goes to when you're writing up, when you say things to do like quantities, 200 milligrams, there should be a space between the 200 and the meat, okay? Or standard. I've seen some of your, when you're writing it up, some of your, there was no space in between. One of your, like number 2 there.
Okay.
Yeah. And then in presentation, we don't say figure 1, figure 2, figure 3, unless they are all in the same slide. Okay. In terms of manuscript is okay. In publication, you can write figure 1 and refer to it. But in presentation, just say the name of that figure. Don't number them up.
Okay.
What was the abundance unit? The abundance unit.
It's in numbers.
Which unit? Yeah.
It's numbers. It's percentage.
Percentage.
Yeah.
So you could just have written abundance in bracket.
Okay, percentage.
So that somebody is able to relate that up.
Okay.
And I've given you comments in the morning. Please, at least you did well by looking through the audience, right? That is why you have a second screen there. This is our screen. Your screen is in the PC. Look at the screen briefly, take a break, look at the people, look at what they are listening to, because the moment you started reading through your methodology and so on, everybody was taking out their phones to start chatting with their friends. That was not good. It tells you that maybe the crowd is not interested in what you're saying. So once you're able to engage with them physically, but don't include them, you'll see that they're not interested, and then maybe you kind of speed up or try and change your tone or something else. Okay, but overall you did well.
Thank you.
From the platform, from the group, yes. Mine is
just a question. I would like to know the species of the aloe vera that you used. Because
Okay,
we have many varieties.
Okay, it's Aloe barbadensis.
So how did you know that it's the one that you just said?
Based on the distribution.
I thought you guys did some molecular studies. Were you able to identify it up to that level? We're only using morphological identification to do that.
Yeah, so based on what we were provided and based on what is found here, so we assume it is Aloe barbadensis.
You can't assume. In science you have to have data, right? So tell us, maybe you used morphological ID to say that up, or use molecular tools to...
Morphological.
Exactly.
Yeah.
Any other? High school. You know, ice is relative. When you say high school, what did you mean? High school, methanol or something?
High school meaning it was methanol was placed in a flask, then cooled under a certain temperature.
You know, like when you're using, let's say, ice water, okay? The temperature might go up to 0, but if I add sodium chloride, it might go up to -10. If I add CO2 to it, they'll all be ice cold, but at different temperatures. So you need to be specific on what the water part that you use to carry out your analysis. Then something else that kept on slipping your tongue was HPTMS. Somebody had that somewhere in the group, right? HPTMS. What is that? HPTLC, it's a chromatograph. There was no MS. Okay. Yeah, but that's fine. I think they did a good job. I wish to welcome our second last. Give them applause for their sitting down. Wow, now we have a teen electrical group. Please prepare as many questions as possible to this group. Okay. And if you feel like sleeping, alert your neighbor to pinch you so that you still remain with us.
Hi everyone, I'm Nikki Ramirez, and this is Duncan Chalo and Benji Omale, my partners. And we're doing Torreya Nilotica. So an overview, quick overview, we'll be doing the introduction, the materials and methods, the results and discussions, conclusion, acknowledgements, and then we'll go through our references. So for our introduction, Torreya nilotica is a shrub or a small tree, commonly known as the bushveld honeysuckle tree. It's found in these eastern African countries on the map there, and it's mainly found on the coastlines. It's also part of the Meliaceae family, and it produces flowers and fruits, as seen in the pictures. So quick background, the Meliaceae family is rich in various secondary metabolites, such as limonoids, sesquiterpenoids, and triterpenoids. And studies have found that the leaves have been used to treat stomach aches and malaria, and the roots have been used to treat headaches and indigestion. And the limonoids and protoliminoids found in Torreya nilotica have been used for various purposes, for medicinal purposes, antifungal, insect antioxidant, and plant antiviral activities. So the purpose of our study is, first we collected leaves, bark, and twigs from Machakos, Kenya, to determine the secondary metabolites in the plant. And these secondary metabolites are important because they are valuable medicinal properties. They have valuable medicinal properties that can be extracted from the plant to further medicine for all to use.
And yeah.
With the sample collection, this is us collecting it in Machakos. Our coordinates are there. It's 1,260 meters above sea level. And this is a water specimen. Our plant is quite interesting. And what we collected the leaves. And then we had twigs. Twigs are small branches, which we also collected, and also collected the bark.
Thank you so much. I think everybody has been talking about the methodology. Of course, those are the steps involved. I would just like to comment that when we started doing the work, we had issues with developing the TLC. It was having the HPTLC. It was having some extra line because of the reagents, the solvents. The toluene and ethyl acetate gave us a lot of problem. We had like 4 or 5 HPTLC before we could go down and have one nicely developed process. So I won't talk much about that because everybody has spoken about it. So just move forward. Move again. Yes, for low polarity, medium polarity, those were the solvents, toluene, ethyl acetate, we worked on it, and then diethyl ether and tetrafuran, water and formic acid. So I won't dwell much on that. HPLC method, we label our sample here, Rea nilotica. So if there are leaves or twigs or the barks, everybody, we all went through this process, 200 milligram, in 1 ml of methanol for GC, and dichromethane for liquid chromatography. That was same process for everybody. We ran our HPLC, and then go back straight to the results.
Okay, so the GC methodology was very similar to the LC methodology, except we labeled it GC. Yeah, that's pretty much it. And then for the parameters, these were the parameters for our GC. We injected 1 microliters of the volume of our sample into the GC for it to run. Yeah.
So this is a peak of the HPTLC. We try to develop the fingerprint. We can see the leaf. This was the leaf, and then we had this low polarity. So I think on this slide, the most important thing, we try to do the antioxidant activity, because we can't get much on the TLC fingerprint apart from just showing that yes, we had some compounds that we could able to see their separation. But the most important thing with the HPTLC that we were able to detect antioxidant activity. Here we did DBPH, this in vitro kind of assay, that we saw there was a quite significant antioxidant activity on this particular, and we used the medium polar to actually develop the HTLC on antioxidant activity.
With respect to the LCMS data, this is quite interesting. From the back has been done previously, and it's important to note that the back was collected from Mombasa, Diani, but our collection was from Modebeli, quite different, 0 degrees, at least around 100 meters above sea level. Ours is 1,200. You can see 3 compounds. One of them is nitroxine acetate, bisphenol A, and isotine. This from the back, as we have them there. It's interesting to see the twigs. We also got minisodium acetate as from the back, and then we had bisphenol. But it's important to see that there are many other peaks that we were not able to detect. Perhaps this is one of the things that we can follow up. On the leaves, interesting leaves has also been worked on, and they never reported about these protolimnoids. What is being reported previously is stigmasterols and the sterols, the cystosterols. But we got milticin, we got these two napubicin. This is the first report in the leaves, and we also got bisphenol A as the back. So I just overview about a good graph, a good table to show what is what and what is where. This is what we got from our study. Niloticin from these plant parts. This is whether the compound is reported in other studies, where it is being reported, whether it's the same plant. This is the same plant. Sometimes for this compound bisphenol A, it's been isolated from another member of the Meliaceae family, which is called a genus Alpha Maxi. We also have tunabicin, which is also reported from the back of the same plant, but also another genus. Quite good activity with reference to reports, antibiotics. You can see very good anti-inflammatory reports being reported. Cytotoxic activity that is quite interesting, less than 20 micrometers. And those are some of the few biological activities we got.
Okay, so for the GC results, in the leaves, we had 36 total compounds, and these were the 6 most abundant ones. I'm not going to read them all. And then for the twigs, same thing. We had 22 total compounds show up on our chromatogram, and these were the 7 most abundant that showed up. And then for the bark, unfortunately, our GC results didn't come out great. So we think it's because we didn't have enough solution when we ran it on the GC. So maybe next time we could try to get more solution extracted. And so for those compounds, total there was 8 that were relevant between the two, leaves and twigs. And they all have their unique medicinal uses. A lot of them have anti-inflammatory medicinal purposes when you use them. Some of them can treat tumors, some of them can treat malaria, some of them are antifungal. So these are really great compounds to extract from the leaves and twigs of Torreya nilotica. Yeah, and then conclusion.
So a total of 36 and 22 compounds from the leaves and twigs were marched to the library. So this is the first report on secondary metabolite on the twigs. No study has been done on the twigs. The protolimnoids were identified. That was niloticin acetate and bisphenol A. And then on top of that, these were the first reports for tunabicin F in the leaves of the T. nilotica. Then these compounds have been found to have a range number of pharmacological activity as reported. He has mentioned cytotoxic effect, anti-cancer. Our interest here, I think we should do more, we should work more on this plant because the sesquiterpenoids have been reported in other plants like the Artemisia annua, which have the antimalarial activity. So it will be, I think, good for us to proceed on this particular work.
So, what is our recommendation? We need, as you saw, we have some peaks, we have compounds for members we did not really identify them. So we can also extend the study to the roots. We can do replicates on the back, as you saw, one of our experiments backfired. We know the history of protolimnoids and limnoids in Meliaceae for their insecticidal and pesticidal. It is of importance not to note that from this plant, not even the extract has been investigated for such activity. Further anti-cancer activity can be done. And then we can also investigate the genus Torreya because we have in Kenya 13 species of Torreya, and globally we have 67, and I think it's only 1 or 2 that have been studied, so that there's room for more to be done.
Okay. Real quickly, acknowledgements. Special thanks to all of these people and yeah, okay, sorry, I lost words. References, all these references, and thank you. That's it.
Yeah, that was really nice. Yeah. How did you feel about this presentation? It was good, right? Yeah. At least you guys have done justice to that.
Good for real, for real.
Yes. It's good. But of course, we are all here to learn one or two things, because nobody, immediately you finish learning everything, you will be in a different dimension of what you go somewhere else. But if you're still in art, we'll still continue learning all through. Okay? So that was really nice. Maybe I'll just give you a story how they discovered neem as being an insecticidal plant. During the swarm of desert locusts, it swarms in billions, okay? As it was moving across, they realized it's only neem plant that was surviving. Huh? That was the only green thing that was left standing. So a couple of guys saw, why is it this particular plant not being eaten? They decided, maybe we need to identify what is present in it, and they ended up identifying it as compounds that have some insecticidal plant. So to your case, what do you mean by anti-inflammatory?
Anti-inflammatory, the compounds have been reported to ease inflammation. When there is inflammation, it's associated with pain, and therefore an anti-inflammatory reduces muscle swelling, and therefore the pain comes down.
I wanted you to explain based on your pharmaceutical knowledge. Use the real terms. Those of the prostaglandin hormones, the EGO, talk to us because that is your area. Okay?
I wasn't prepared for that.
Okay, that's fine. So we really appreciate the only maybe a point that they kind of confused us a little bit is they were using sodium anhydrous, sodium sulfate in the HPLC preparation. You saw that, huh? You saw that. So what happened?
I copy pasted it and I forgot to delete it.
Okay, now back to the floor. Anybody else who wants to maybe give a comment or a question? Or we're all sleeping. Maybe I think we need to stand up for some 3 seconds or so. You have a question?
So you have at last you have a anti-pesticide activity. What's an anti-pesticide?
Where?
Probably a little bit early. After this. Or you are recommend you see that the second item, anti-pesticide. What's that?
Oh, sorry, I need to remove the word anti-pesticide. I understand.
Also have an anti-mosquito activity.
Yes.
So I'm not sure people
Okay.
use anti-mosquito or you want to use another process.
Maybe depending on what stage you're working with, if it's lab or maybe vaccine activity, depending on which stage you're working on.
Yeah,
but
so
we can develop it further to see.
Yes.
And I like the comment which was given by Andrea about when you see that profile in terms of this analysis, know very well that the syringe just picked up air, it didn't pick the solution. That is very correct. Okay? And I'm happy you brought it out. Yeah. Any other comments?
So maybe
What happened to the backbenchers?
Yes.
Okay, that's fine. Yeah, you want to say something?
Yes, the reason why we put insecticidal is because, as you can see, one of these compounds is having a very good activity against Culex mosquitoes specifically. And then also from our knowledge of limonoids and pesticides, which anti-mosquito cannot necessarily mean anti-pesticidal. So it is, is from
not anti-pesticidal. Remove that term from your...
Okay, the pesticidal activity will not confirm does not necessarily mean insecticidal.
Okay, we give them a round of applause. So far we have done so good, but very bad on time. I wish to welcome the last but not least group for this session to give us a talk.
think you understand the same. It should not be USDC, it should be USDC. Small letters.
Yes, we trying to sort out the slide issue. It's not an opportunity for us to make noise, okay? You're only allowed to talk if you have a question to say. Maybe I can allow you to look into your phone because that is silent. Because if you talk, you're kind of distracting everybody else. So what is DPPH? From your, I saw most of you, all of you are just saying DPPH. What is it and what is it used for?
You should be able also to explain the mechanism.
Sorry? No, DPPH for the antioxidant activity.
I assume that it's an oxidant.
It is?
It has to be an oxidant if it's an antioxidant acid, right?
So what does it do?
Some kind of free radical.
And then it binds to.
It has to be a general.
What I want is somebody to explain the antioxidant activity that you guys did in HPTLC and other.
Then it transforms.
So we are all comfortable that we don't know, we know, but we don't know how to explain which is which. Do we know what's whatever we're looking for? Where is the guys for Turea group? Maybe they could tell us the DPPH, what it is, and the antioxidant activity.
It's 2-diphenyl 1-pyrazide. It is scavenges. It's a kind of free radical. So it is when there is compounds that scavenge for free radicals. So our compound, for example, was able to scavenge for free radicals, and therefore the diphenyl pyrazide, the 2,2-diphenyl pyrazide changes color, and that is used to assay the oxidant activity, the antioxidant activity.
Clap for him. So, in my culture, we have a saying that says, nature obeys no law. Have you ever heard of that? Nature obeys no law. So I'm giving you a 3-minute break. Let's go on stretch. People want to go to washroom as you're setting up the slides. Okay, you're free to make noise, go on stretch a little bit and so.
Is it human nature to rebel? Because I've given you 3 minutes to go and stray, do other stuff, and then you're just rebelling against it.
To be honest.
Oh, you were good.
I think it's for
amejiwekelea mpaka anashangaa kwa nini amejiwekelea
But I follow a lot of concepts.
Kiongozi, unanirekodi?
Your tea tree oil is the center of this. This is where Eres was born, because we were only thinking about tea tree oil before any other plants came. So expect a lot of curiosity from everyone.
Please do.
For the purpose of time, because we still have 2 more other programs this evening, unless you want us to give you certificates next year, I will ship it to you guys. Now we can do a Zoom meeting and hand over some copies. We still have poster session, it takes time. So because of time, I will wish us that we just finish up the last presentation, we give them due respect because you are also quiet during your presentation. So please be quiet, listen to them, pay attention. Where you can improve on their presentation, please do so. If you have a question, raise it up. The experts around the room, they'll be able to give you some feedback. Okay, so T3 group, the floor is yours. Please kill it.
Oh, hi everybody. So this is team T3 Oil, and we're going to present to you our findings on Melaleuca altifolia.
My name is Manisha Shelby. I am at Howard University in the Department of Genetics.
And my name is Betsy Alice from the Department of Biology, University of Nairobi.
Today we're going to cover first the introduction, then we'll go over a brief literature review, followed by methods. The results and discussion we'll cover briefly because we all pretty much had a lot of the same methods, but our results are kind of distinct. And then we'll finalize it with the conclusion. An introduction.
Okay, so why is ethnopharmacy relevant? Ethnopharmacy is going to help us to do research on various plants so that we are able to identify the bioactive and therapeutic constituents to facilitate drug discovery.
And the main objective of our research was to actually determine the presence and relative abundance of these secondary metabolites of the leaves of the Melaleuca alternativa plant.
Okay, so onto our literature review. This is actually, it's not so clear, but you can see it. It's actually a Melaleuca alternifolia tree. The tree actually grows from 3 to 7 meters tall. So this is it. So, Melaleuca alternifolia is an evergreen tree, and tea tree oil is an essential oil that is extracted from this tree, the leaves. It has puffy white flower clusters, as you're going to see on the next slide, that are near the top of the tree, which attract bumblebees and other pollinators.
And something I learned while during this project is that the plant is perfect, meaning it has both male and female components. It is a family of the Myrcidae, and it is the common name is the paperbark tree. It has really thin, leafy bark, and the name Melaleuca is kind of contradictory because Mela is the dark and Luca is white, so those white leaves and the dark black bark. And as Betsy mentioned, it has those nice fluffy white flowers which bloom during the spring and summer.
So, I'm going to tell you how tea tree came to be used in medicine and how we are able to use it. So the Aborigines in Australia are the first people who were able to discover tea tree oil. And then, so this man is called Captain James Cook, and he was making a famous trip with his mentee, Joseph Banks. So he was able to collect the tree, and he was able to brew a spicy and refreshing tea from the leaves. So from that, he was able to give the name tea tree oil because of the scent. Okay, so from then, the samples were taken to England for further studies, and in the 1920s, Arthur Penfold, a chemist, from the Sydney Museum of Technology, was able to do experiments by extracting oil from the leaves.
And that started the beginning of the commercial use of the tea tree plant. However, it had been used in Australia for many of years by the Aboriginal people, and the grounds where these native people were found, it's called healing grounds to this day. The leaves were crushed and applied directly to the skin to actually heal any topical ailments. You could infuse the leaves in the water and brew it for sore throats and any type of sinuses that you may have, and it could be inhaled from the smoke so that you could heal the coughs or any colds you may have. And to actually preserve the plants, because we didn't want to actually get rid of them all once you're trying to harvest them, it's harvested by a process called coppicing, so that the trees will grow back. So during the winter time, they'll go ahead and cut close to the bark so that the roots can maintain themselves and grow back and replenish.
Okay, so during the cultivation, that grown in both in Australia and also here in Kenya right now in Lunga Lunga, we have a plantation of tea tree oil. So during the harvesting, distillation is done. Steam distillation is done on the other sites. So that's how they're able to extract the leaves. So it has been found to increase membrane permeability. It disrupts cell membrane functions and inhibits cell respiration.
So some of the major compounds of tea tree oil are the alpha-terpineol, terpene 4-ol, and terpenol. So inside of these components, you'll find a lot of antimicrobial, antifungal, antiviral components, even anti-cancer components. Because most interesting, what I found is it could be used to treat melanoma cancer, which is one of the most persistent cancers, and it does this by resisting the apoptotic ability of the cells. And you'll also see some people using it as a douche so that you can wash out your throat as well for the vagina as well. Also, as I mentioned before, the antimicrobial properties of the plant, it has been known from the International Standardization Company to have these certain concentrations so that it could be used in commercial uses. So the alpha-terpenol, you have to have it at least 35%, However, the 1,8 cineol cannot be used more than 15% because it actually prevents the antimicrobial properties of the plant, and it's a skin irritant. So we'll move on to materials and methods, which we all kind of shared with those methods. That includes sample collection. We went out to Machacos, but however, our plant was not found there because it's exotic. So we had to wait for someone to actually go to pick up and bring us back our plants. And we did not get the actual coordinates for those, so we apologize for that. But after sample collection, we were actually able, well, I actually helped with a lot of you all's plants with the pressing process. If you're gonna be ready for that. We did a lot of the extraction. We used methanol for the extraction. We did GC, HPLC, as well as HPTLC analysis, as well did. And here's a map of where Thika is located within Kenya and the sample collections.
Okay, on to the sample collection. So first we were able to get a specimen that was already grown, basically, but this is a summary of what happens in the field. So when you identify the specimen, the fertile specimen, meaning it has the leaf, the flower, and probably the seeds. So you get to record the common name in the field, the GPS coordinates, and the altitude of the place, including the ecological conditions. And then you do the pressing by mounting on a newspaper between the pressing boards, and sterilize the plant parts, then have them in tertiary packaging. That's the Ziploc bag, the red bag that we had, and the kind of stuff that we had. You'll see them in the photo. So this was our trip to the field collection. And as you can see, Shelby is actually noting down the GPS coordinates on the newspaper before she gets them on the pressing board. So that's what we were using to record the GPS coordinates. Yeah, and it was so much fun. And this is the Ziploc bags we're talking about. So this is the primary packaging. Then we have the secondary, I don't know if you can see it. And then this was the last packaging that we were using.
But not our this was for you all, but just to give you an example of what it looked like. And there's an extraction process which took place at the sea base, which was done actually with methanol. We had the actual protocol that you all shared as well. We said about 200 milligrams of the leaf sample, which was crushed and ground really well. We added 1.5 of the solvent, sonicated, centrifuged, then filtered that specimen, and we prepared it for all of the analysis that was to follow. The gas chromatography followed by the mass spec parameters. These were some of the things that were shared amongst all of us with the temperature regulations, pressure as well. And then with the HPLC was performed, following those same methodologies. And then the HPTLC, which I think was so kind to share with us, which we followed these instructions. And the derivatization process, we had the mobile phase, stationary phase, which we all kind of went through with that really expensive equipment that was given to us. And these were the reagents that we used for the derivatization. And now to the meat and butter or potatoes.
So we were able to do the GCMS analysis and top on our list with these compounds. So we were able to identify specifically sabinin hydrate. It's anti-aging for the skin. So that was a highlight for us. We also identified terpene for all, bicyclogermal cream, stearyl alcohol, and camta. For LCMS, we were able to identify stigmasterol, generally linalool. Actually, these are the ones that we were able to identify. Yeah, so when we did our HPTLC,
these were separated by polarity, and a lot of the terpenes are going to have the low polarity, so that's where you'll see the high concentration here across the top. And those that were with the hydroxy groups, that would increase the polarity. So those are the things we switch towards the bottom. Those were the components that are mainly known for their antimicrobial components, antifungal components. So that was going to be really high for our tea tree oil and the commercial uses. And this plant has very, very high antioxidant activity, as you can see both in the white light as well as for the UV250 full spectrum. So here's a pie chart of everything that we saw. And the main component where you see almost at 20% was the terpene 4-ol, which has a lot of antimicrobial activity. And this is the actual component that you want to see a lot of in those commercial components, in the commercial use. So in conclusion and future prospects.
Okay, so at top of our list we have this compounds that we were able to identify from all the three analytical methods. So that's GCMS, HP, LCMS, and thin layer chromatography. So if you see, like I told you, sabinin hydrate is anti-aging, and you've just noticed that our plant had a very high level of antioxidant activity, courtesy of terpene 4-ol. Yeah. So bicyclojamacrine has anti-inflammatory activity. That's just a summary of what we have.
You do not want to ingest tea tree oil, just to let you know. However, overall, the tea tree plant is safe for use, but do not ingest the oil. And we saw from our analysis and our results that there are a lot of other things where we should be researching on to see what could we use this plant for as far as therapeutics and medicinal purposes. And that concludes our presentation.
That was really nice. Give them a round of applause. We have done our last talk to be as good as best. So I really enjoyed the talk. There's some maybe point of improvement that I'll come last. I'll give 3 opportunities from the floor to comment. These are your colleagues, these are your fellows, please. Yes.
You guys use commercial tea tree oil in your analysis, right?
So because the commercial actual component, it was diluted so much, we didn't get a lot of results from it. So we actually used the leaves for the HPTLC. But that was a good catch because, yeah, we were trying to do the commercial use of this, but we do have this to pass around for you all. If you would like to pass that around and take a smell and see why the IRS.
What do you mean? You didn't use the commercial one, right?
So we used it, but because it was diluted so much.
We used it, but it was too thick, so we were not able to get the separation.
Too thick or too diluted?
Too thick.
So once we used it was thick oil, but I think we diluted it so much. Yeah, it was diluted so much that we didn't get enough of it.
Diluted from the market or you? They diluted.
Okay. We were doing the extraction, and we were using ethanol. So after the extraction, when we did the analysis, we were not able to see the separation clearly. So we were just trying to find out what the problem was, and probably it was the extraction.
Yeah, that's your error, not. Okay, I get it. Any other comment from the normal floor? Okay, go to any other comments? You're good? Okay, Haitebo.
So probably asking something you may not know, since for its TJ, there are some adulterant. So to determine the adulterant, European Pharmacopoeia and the ISO have a standard, so they have some quality. So for this result, do you think this teacher on your
would fit the standard?
Yeah, is a good one or not qualified for?
So because our terpene 4-ol is only at 19%, and they recommend it to be at least 35%. I would not use our extraction method for any commercial uses, no, but if you give us another year to come back, we could perfect this method and we could go into the commercial industry with our extractions.
Thank you for the nice presentation. Did you test your products, the market products, with all the methods that you use, the LCMS, BCMS, and
No, ma'am. So we only, because again, we were not able to actually even extract our plant from the field work that was done. and the commercial products were provided for us only at the time we did the HPTLC analysis. And that's the part we were talking about, that we made the error of over diluting the actual sample.
Okay, that's nice. So I'll just give you now maybe, for the university, this the headings are okay, objective, introduction, and so on. But for big stage, stop using the traditional names, yeah? If you're talking about literature review, you can just say what is known. You know, that will be so catching. People will be interested. What is known about this plant? Okay, the university, you know it as maybe a literature review and so on. So try and avoid those, the terms for general audience. Next is about your structures. Even your images, when you want to get clear images when you're expanding it up, you don't pull from the middle. This is the area, when you pull from the middle, you'll end up having structures that are blurred, that are out of shape. But if you pull it from the sideways, you can actually expand the structure to fit the whole page. So this for all of us here. Go back to your pie chart, just mention a few.
Which one?
The pie chart, the big pie chart. You see, these are chemical terms. You all know the symbol for gamma is, use the gamma symbol, use the maybe the C is C, trans is E. Those are standard. You know, when you're giving to audience, even if you're not a chemist, they'll know, yeah, they did some search on how to present the molecules on that. Okay, so I heard you mention about they normally bloom during spring, summer. We don't have that in Kenya. So please comment something.
So, and that's funny because I actually looked to see that it was going to sprout during the November and December, which will be our winter in the northern hemisphere. So I figured because it's winter here right now and it's summer.
And it's cold season, it's not winter.
So is it like wet and dry season?
Yeah.
Okay.
So when do they flower in, let's say, Africa? Okay, maybe
November, December.
Yeah, around the equator, right?
November, December.
December. That is what should come out clear because these are different audience. So when you start talking about winter, summer, autumn, and so on, we don't know that.
No.
Okay. Anything else? I think we need to give them a round of applause for this. Now hand over to Professor Sheila to take over to the next session.
So we can break for tea. I can go for tea and then we come back for the last session.
Good job.
All right, so let's break for tea, then we come back for the last session. You can clap for yourself as you go for tea.
How was your presentation? It was great.